A3. Well, fantastic. It's been really amazing to see all the interactions with the scientists and attendees. Heard a lot of lively conversation, so let's hope to continue that throughout the day. Let's get started. Really quite thrilled to host Hongkui Zeng, who is the Executive Vice President and Director of the Allen Institute for Brain Science. So thank you for coming over from across the country. Dr. Zeng has been studying neuronal diversity and connectivity in the mouse brain circuitry for many years. She has a well-known pedigree in neuroscience where she received her PhD in molecular and cell biology from Brandeis University and postdoctoral training at MIT in neuroscience. She has contributed to a huge, I would say, universal adoption of a community resource in the mouse atlas. Anyone who's ever studied brains have probably used it, so I think it is fairly universal, so huge contributions, and we are so thankful for that. She's received so many awards, including the recent 2023 PADEL Research Award from the National Academy of Sciences. She's an elected member of the National Academy of Sciences and the National Academy of Medicine. And we are really quite pleased for her to be able to share her talk on the cell type and diversity and organization of the mammalian brain. So, thank you.
Okay, thanks everyone. First of all, I want to thank Catherine for that very nice introduction, and thanks for the invitation from especially Evgeny for attending this conference. This is my first time at a conference. It's great to learn about all these exciting new imaging technologies. Okay, so today I'll tell you about our work at the Allen Institute for Brain Science in understanding the diversity and organization of cell types in the mammalian brain. The collective goal of neuroscience is to understand how brain works at multiple levels, from cells and circuits to computation to eventually the generation of behavior and cognition. This is a daunting task given the extreme complexity of the brain and the nervous system. With millions to billions of neurons and a similar number of non-neuronal cells in the brain of all these different amazing species with amazing behaviors, and the thousands of connections each neuron can form with others. To tackle this complexity, we think that it's essential to take a multi-level approach from cell types to connectivity or wiring diagrams, to in vivo physiology, and to eventually, hopefully, a computational understanding of how brain processes information and generates behavior. At the Allen Institute for Brain Science, our focus has been mainly on the first two levels, understanding diversity of cell types and how they are wired together to form functionally specific circuits. We use a team science, open science approach, creating large-scale technology platforms and pipelines, using them to generate really comprehensive data sets and present the data as foundational resource to the community. At the same time, we also use a data-driven approach to discover organizing rules and principles from the data regarding the organization of the brain at cell type and connectivity levels. So, as we all know, ever since the age of Ramani Cajal more than 100 years ago, there are an extraordinary diverse array of cell types in the brain, and they have really tremendously heterogeneous properties. For example, the morphological and projectional properties is beautifully illustrated by those drawings by Ramani Cajal himself. But not only that, we now know that the cells also have diverse properties at multiple other levels, from molecular gene expression, proteins, distributions, to physiological properties and connectional and functional properties. So here are the set of approaches that we have established at the Allen Institute in profiling single cells and deriving cell type taxonomies from those data from single cells. So our front-end approach is the highly scalable single-cell genomics technologies, including single-cell RNA-seq or transcriptomics, single-cell epigenomics, and mainly really ATAC-seq, and spatially resolved transcriptomics at single-cell or near single-cell levels. And we use those approaches to profile hundreds of thousands to millions of cells, and through computational clustering to create the first iteration of cell type taxonomy. And then we create genetic tools utilizing the information gained from single-cell genomic study that will allow us to label and manipulate specific cell types in the brain. Traditionally, we have been generating transgenic mouse lines, driver lines, using marker genes identified in the data. In recent years, we have switched to a more versatile approach, which is to identify putative enhancer elements through the combination of single-cell transcriptomics and epigenomics data, identifying cell type-specific putative enhancers, and put them in AAV viruses and test their specificity. And in a pretty good success rate, we have been able to identify hundreds of enhancer elements that can drive cell type specificity in the brain. And not only in mouse brain, we're now able to apply those enhancer AAVs in non-model organisms as well, such as the non-human primate brains and even the human brain itself for investigative studies, as well as for potential, the development of potential therapies. On the anatomical side, we use large-scale light microscopy and electron microscopy to obtain the brain-wide morphological and projectional patterns of individual neurons, as well as dense local circuitry reconstruction to obtain the local morphologies and their synaptic connections with nearby neurons. To bridge molecular information and anatomical information, we use a set of multi-model approaches, including this triple modality patch-seq, in which you patch an individual cell in brain slices. You record the electrophysiological properties from the patched cell. And then you also fill the cell at the same time to obtain their morphologies. And then at the end of recording, you extract the RNA content from the cell and obtain single-cell transcriptome from the recorded cell. So using this kind of approach, we're able to obtain morphological, electrophysiological, and transcriptomic information from the same cell and use that to define cell type in a unified manner. We also use bar-coded connectomics approaches such as MAP-seq or BAR-seq, again to relate the long-range projection patterns as decoded by in situ sequencing of the barcodes with the transcriptomic profiles of the barcode labeled cells. We also couple traditional anterograde and retrograde tracing with molecular profiling such as retro-seq or retro-Murphish to, again, relate transcriptomic profiles with long-range input-output projection patterns of individual neurons. Underlying all these different technology platforms, we build strong computational capabilities for not only management of all these different types of data, but also for analysis, especially integrated analysis. And now we very much incorporate and wanted to incorporate more of AI and machine learning approaches into the analysis. And finally, we also build online platforms, visualization platforms to disseminate the knowledge, the data, knowledge, and tools we derived from all these different studies. Okay, so with that overall introduction, now I will tell you about a series of studies that we have done in understanding cell types in the brain. And our starting point, as I said, is using single-cell transcriptomics. So I will start with that too. So, and we started with single brain regions. And the first study is actually a collaboration within the Brain Initiative Cell Census Network, or BICCN. In this published work from 2021, we collectively use the approaches that I mentioned in the previous slide to profile cells from a single brain region, the primary motor cortex, across 4 mammalian species, from mouse to marmoset, to macaque monkey, and to the human. We derived this consensus taxonomy or atlas of all the neuronal and non-neuronal cell types in the primary motor cortex. And what we found was that cortical, and this is consistent with many of the other studies published around the same time about cortical cell types. We found that cortical transcriptomic cell type taxonomies reveal a hierarchical organization of the cell types, as shown here, and the coexistence of discrete and continuous variations, as you can see in the transcriptomic profiles, as well as the cellular properties in other modalities. And also the coexistence of similarity and difference across species. The discrete variations and the similarities are usually displayed at the higher branches, the major subdivisions of cell types, whereas the continuous variations and the differences or heterogeneities are mostly observed at a cluster level, which shows a complex cell type landscape. In a next set of studies, we compare the cell types across different cortical areas. And we find an interesting dichotomy between the 2 major classes of neuronal types in the cortex. Namely, the glutamatergic excitatory neuronal types are highly distinct between different cortical regions, whereas the GABAergic inhibitory neuron types are shared across the different cortical areas. And this dichotomy, we hypothesize that this dichotomy is probably due to a differential developmental origins of those 2 major classes of neurons, with the glutamatergic neurons generated during development within the cortical plate itself and subject to this morphogen defined cortical gradient and regionalization. Whereas the GABAergic neurons were generated in the subcortical or subpallium ganglionic eminence areas that migrate into the cortex during development, and as such, their transcriptomic profiles may not contain this kind of a regional specific information, the intrinsic regional specific information. Okay, so then in yet another study, following on that, we further scaled up our profiling to cover the entire cortex, isocortex or neocortex of the brain, as well as the nearby archaeocortical regions called hippocampal formation. And in this much larger scale studies with 1.2 million cells profiled, 30 different cortical regions, neocortical and archaeocortical regions covered. We identified a parallel organization, especially of the glutamatergic neuron types, between isocortex or neocortex and hippocampal formation, and homologous sets or subclasses of glutamatergic neuron types, suggesting an evolutionary relatedness between those 2 major brain organizations, and a similar parallel circuit organization as well. Okay. So with that summary, then I move on to our latest work, again, within the large collaborative consortium of the BICCN. And in this work, which was just published at the end of last year, this is really a tremendous effort in scaling up single-cell genomic approaches to cover the entire mouse brain and generating this high resolution cell type atlas of the whole brain, which is really the first time for any mammalian species. So I'm very proud of this work coming out of this consortium, especially the 6 groups named here that spearheaded the generation of this large-scale whole brain data set of 3 modalities. single-cell RNA-seq and single-nucleus RNA-seq from us and from Evan McCaskill's group at Broad. And a single-nucleus methylation-seq and single-nucleus attack-seq, whole brain scale, from two groups at San Diego, Joe Ecker's group at Salk and Bing Ren's group at UCSD. And finally, 3 sets of whole brain scale single-cell transcriptomics data set. We at Allen using the Vizgen Merscal platform, Xiaowei Zhang at Harvard using her own custom, the Murfish platform, who, as you know, Xiaowei is the inventor of Murfish technology, as well as Evan McCaskill, again, using the slide seed approach that his lab developed. So with all these different data sets together, we are able to create multiple versions of the whole brain cell atlases. And though importantly, those multiple versions can all be aligned together, mainly using the atlas that we generated at the Allen Institute. So all this other data were able to be mapped to show high degree of consistency with our data. Okay, so then in the next, I will mainly just focus on the work that we have done at the Allen Institute in using those 2 whole brain scale data sets to create this high resolution cell type atlas. So which is shown here. The entire atlas is shown in this UMAP with 4 million individual single cell transcriptomes. And I'll tell you a little bit about how this atlas was created. We first generated 7 million single-cell RNA-seq transcriptomes using the 10x platform. We performed a very stringent QC to fail 3 million out of the 7 million single-cell transcriptomes and retain only 4 million really high quality and consistent quality single-cell transcriptomes. We did that because through the process, we realized it's actually to create a reference cell type atlas, it's essential to keep only the highest and most consistent quality data because then it will allow the mapping and integration of the subsequent data sets. For example, the Murfish data set that we're going to map into later, as well as any other kind of RNA-seq data set from the community. In order to obtain accurate mapping, the reference atlas really needs to be high quality. Otherwise, as you can imagine, if the reference atlas contains some low quality cells, your mapping result will not be accurate. Okay, so with that, we're using the 4 million single cell transcriptomes from the entire brain. We performed iterative clustering using the algorithms that generally used in the field. And we obtained more than 5,000 individual transcriptomic clusters. And from those 5,000 transcriptomic clusters, we derived 8,500 differentially expressed genes that distinguish all those 5,000 clusters. And then from those 8,500 marker genes, we computationally chose the top 500 marker genes that collectively have the greatest discriminating power to differentiate each pair of those 5,000 clusters. And we used those 500 genes for the Murfish experiment. So, and this is from one single brain of a whole brain Murfish data set. We segmented 4 million individual cells. And then we mapped every single segmented Murfish cell back into our transcriptomic taxonomy defined by the RNA-seq data to obtain a cluster identity for each of those Murfish cells. And then conversely, using the collective anatomical location of each of those mapped Murfish cells belonging to each cluster, we assign the anatomical identity for each of those clusters. So through this kind of back and forth integrative process, we now define a cell type atlas, transcriptomic and cell type atlas of 5,322 clusters. And then finally, we also performed a hierarchical clustering analysis to group the clusters into a hierarchical structure because we're not sure if the 5,300 clusters are all discrete real cell types or not. They could, some of the clusters may reflect transitioning states or things like that. We describe the cells at different levels, 1,200 supertypes, 338 subclasses, and 34 classes. And we also organized them into 7 neighborhoods, which are mainly based on large brain structures. A major finding from this exercise is that neurons exhibit extraordinary diversity. Neurons form 5,200 out of the 5,300 clusters that we identified using a uniform, clustering criteria. So, whereas the non-neuronal cells only form about 117 clusters. So, extraordinary diversity on the neuronal front. Furthermore, we find that cell types, especially neuronal types, are defined not only by their molecular identity, for example, by what kind of neurotransmitters they express, be it glutamatergic or GABAergic or dopaminergic, serotonergic, things like that. They are also very much defined by the regions where they come from, as shown here. So in fact, the class and subclass definition shown here is really the combination of molecular specificity and regional specificity. So as you can begin to imagine that the brain is really organized in a spatial plan, as just revealed by the transcriptomic data. Okay, now moving on. We also then analyzed the gene co-expression modules, and here shows 52 transcription factor co-expression modules, and as laid out here. And we find that the transcription factor co-expression modules exhibit beautiful cell type regional and cell type specificity at different levels of the hierarchy, from class to subclass, all the way down to individual clusters. So those transcription factor co-expression modules really collectively form this transcription factor code that defines cell types across the entire brain, both neuronal and non-neuronal. And so in the next slide, we can drill down a little bit further. Here, I'll only show you, the landscape at the whole brain level. But if you drill down, you see the diversity and heterogeneity. You continue to see that, and it's just amazing, even as you go deeper. So out of the 7 neighborhoods that we defined, neuronal neighborhoods, 6 of them are neuronal neighborhoods, and they are grouped by really major brain structures. And we lumped all the non-neuronal cells into the 7th neighborhood. So here it shows 3 neuronal neighborhoods in the forebrain, and they are separated by all the glutamatergic neurons from the pallium or the telencephalic region, all the GABAergic neurons that were generated in the subpallium, or the developmentally defined subpallium part of the telencephalon, and all the glutamatergic and GABAergic neuron types that are in the hypothalamus and the most closely related extended amygdala areas. So as you can see, even within each of those neighborhoods, there are thousands of clusters, hundreds to thousands of clusters, and exhibit extraordinary diversity, as well as the spatial specificity. So each of the clusters is associated with, a specific spatial location as beautifully revealed in the Murphy data set. And then we can go down a little bit further using the subpallium GABA neighborhood as an example, and shown in the next slide. So this is this neighborhood contains all the GABAergic neuron types for the entire telencephalon, including cortical structures, the subcortical structure we call the cerebral nuclei, anatomically, and that contains all the major striatal regions, dorsal ventral striatal regions, as well as the pallidum regions. So underlying that anatomical complexity, we see the cell type complexity, and with many different known migratory routes of all those GABAergic neurons. They form 7 classes, 52 subclasses, 284 supertypes, and more than 1,000 individual clusters. And organized is really well related manner, and also matching very nice with the spatial specificity of the different cortical and striatal and palatal regions. And we discover many new cell types in that process, and also subdivisions of known cell types in this process. And I don't have time to get into the detail. So then, using another example, to illustrate the heterogeneity. And this is to extract all the neuronal types, clusters, that express a modulatory neurotransmitters. So the modulatory neurotransmitters beyond just glutamatergic and GABAergic fast transmitting transmitters. So extracted all of them from the whole brain and put them separately together into this UMAP. And from the UMAP, as you can see that we identified all the modulatory neurotransmitter types, including cholinergic, dopaminergic, noradrenergic, serotonergic, as well as histaminergic. And a very interesting observation here is then that among all those clusters, we observed a heterogeneity at the neurotransmitter co-expression or co-release level. For example, if you look at the dopamine or serotonin, you see that there are some clusters that express. Of course, we don't know about the release. We only know about the expression based on the transcriptomic information. There's the dopamine alone clusters. There's the dopamine and glutamate co-expression clusters, dopamine and GABA co-expression, dopamine and glutamate and GABA co-expression, and even GABA and glutamate alone, okay, so maybe going on to the next. So here I highlight the subclasses that contain midbrain dopamine neurons and midbrain hindbrain serotonergic neurons. So in addition to all these different combinations, we even can observe glutamate and GABA co-expression only clusters that don't even have dopamine, but is still transcriptomically or molecularly very similar to the dopamine neurons themselves. So in the next slide, I further zoom in, just focusing on this particular subclass, 215, which contains only 4,000 cells, single-cell transcriptomes, but covers all the dopamine neurons within the midbrain structures, including substantia nigra, ventral tegmental areas, and the midbrain Raphe nuclei. And as you can see, the heterogeneity shown in this UMAP, those 4,000 cells within the single subclass can be divided into 8 supertypes and 43 transcriptomic clusters. And also, if you color them by their neurotransmitter content, it's highly heterogeneous as well. It's the same kind of content that I showed you before. Dopamine alone, dopamine GABA, dopamine gluta GABA, and all triple, 3 transmitters are shown together. And then spatially, okay, sorry, go back. Spatially, they are also, very often the different supertypes or clusters are very much intermingled together. There is some degree of specificity. For example, this particular supertype is very specific to the substantia nigra region, and not so much in the VTA, or a little bit in the VTA, but not so much in the Raphe nuclei at all. Whereas all the other supertypes are very much intermingled within VTA and the Raphe, dorsal and medial Raphe nuclei, and suggesting their collective roles in mediating the emotional functions and other neuromodulatory functions of the dopamine neurons. Okay. So I also wanted to talk about the diversity within the non-neuronal types. Even though I said in the brain, relatively speaking, the non-neuronal cells exhibit much lower diversity compared to neurons, we still observe very interesting diversity. And I just wanted to use the astrocytes and the ependymal cells as examples, really good examples here to show the diversity. So here is a UMAP showing all the astrocyte clusters that we have identified, and the spatial specificity of each of the astrocyte cluster that's revealed in the Murfish data. we find that there are 3 major groups of astrocytes in the brain. And the first group is the dominant astrocytes that are present in the gray matter of the brain that are intermingled together with the neurons, right? All different neurons. And we find that this dominant group of astrocytes exhibit also great regional specificity, although not as refined as neurons, but they are distinct from each other in all the major brain structures, from telencephalon, cortical areas, subcortical areas, to striatum, hippocampus, and then midbrain, hindbrain, and then cerebellum area. So there's a lot of region-specific astrocytes. The second major group of astrocytes is called interlaminar astrocytes, and those are the astrocytes that line the peel surface of the brain, and they send these long processes from the peel surface into the gray matter. And then the third group of astrocytes are the special ones that may even have neurogenic potential, because they are the ones that line the rostral migratory stream in the brain. And the rostral migratory stream is a special kind of a tunnel structure that allows the newly formed olfactory bulb GABAergic granule cells through the adult neurogenesis process, the newly formed cells from the subventricular zone in the dorsal striatum and migrate along the stream all the way into the olfactory bulb. So in addition to the immature neurons that we can see that's going from the dorsal striatum, subventricular zone, into the olfactory bulb, we also see the series of astrocytes that lining that stream, and the transcriptomic clusters shown that form this trajectory matches perfectly with the spatial transition of the astrocytes from dorsal striatum into olfactory bulb. So another good example of the transcriptomic specificity correlated with spatial specificity. So here is another example. We observed yet another large set of ependymal cells. Ependymal cells are very similar to astrocytes, but they are the special kind of glial cells that line all the ventricles in the brain, different parts of the ventricles, and as well as the circumventricular organs. The ependymal cells play important roles in mediating brain blood and brain and cerebral spinal fluid interactions, gating nutrient and hormone transport across the brain. So we find many different ependymal cell clusters, and each of those clusters has a beautiful specific location in different parts of the ventricles and circumventricular organs. And in particular, I want to point out this tenicyte subclass, which is one subclass of all the ependymal cells, and all the tenicytes are located in the ventral third ventricle within the hypothalamus, and they correspond very well with the traditionally defined tenicytes that have specialized morphologies. Okay, so just to summarize all this work together, with this whole brain atlas, we have now built a new online platform called Allen Brain Cell Atlas, or ABC Atlas, in which we display the whole brain transcriptomic data, as well as the whole brain merged fish data that allow anybody to use and to navigate and to learn. Furthermore, we have built a computational tool called MapMyCells, which is a cloud-based tool that allows a user to upload their own single-cell, single-nucleus RNA-seq data, or even spatial transcriptomics data, any kind of a cell-by-gene matrix, and map each cell of their own data into our whole brain reference atlas to obtain a cluster or cell type identity for their own cells. And they can use that for comparison, and they can use that for compare with their own clustering results and for integrative analysis. We now are adding new computational tools, integrative tools, into MapMyCells platform, just so that people have more ways of comparing their own data with our reference atlas data. Okay, so with that, after introducing the reference atlas, that we have created. I want to then tell you a few additional studies that are unpublished about how we have used this kind of reference resource to further gain understanding about cell types, how they change in development and aging, and how they are correlated with their connectional and possibly functional properties in the future. Okay, so in this study, we profiled, we completed another large-scale single-cell RNA-seq profiling in the aging mouse brain. So in this particular study, we generated 1.2 million single-cell RNA-seq equally divided between aged mouse brain, 18-month-old brain, and young adult brain. And that covers 16 broad brain regions in forebrain, midbrain, and hindbrain, altogether about actually 100 high-resolution brain anatomical structures, about 1/3 of the whole brain. And then we also generated some spatial transcriptomics data to validate the findings that we obtained from the RNA-seq data. To obtain cell type identities of this from this data set, we performed a label transfer using the MapMyCells tool to transfer the cell type identities at class, subclass, and supertype level from our whole mouse brain taxonomy into this aging data. And we also performed the de novo clustering in order to identify aging-specific clusters. So overall, from this data set, we obtained 172 subclasses, about 800 clusters. And we used 3 different computational approaches to identify age-associated differentially expressed genes. And eventually, we identified about 2,500 genes that have changed in some part of the brain between the young adult and aged brain, and a small number of age-biased clusters, only 12 age-biased clusters. So from these numbers, you can see that the transcriptomic clusters do not change dramatically, with age. They don't, often form this kind of discrete clusters. Only in special cases, dramatic changes happen. Whereas with aging, the greatest change is at the individual gene expression level within the existing, defined clusters. So more subtle gene expression changes rather than dramatic, the formation of new cell type or new cluster. So here summarizes the distribution of all the 2,500 age-associated DE genes across the major neuronal subclasses and also non-neuronal supertypes. So here I want to emphasize that even though I said that there is not as much diversity of the non-neuronal cells in the brain, but here is a great illustration showing that actually the non-neuronal cells in the brain change dramatically. So they change a lot more than neuronal types, with age, with other possibly other diseased conditions. So we see a lot more DE genes associated with non-neuronal types. That's why we show it here at the supertype level, whereas the neurons, we group them at subclass level. So you can already see hotspots of changes on non-neuronal cells, but also hotspots of gene expression changes associated with cortex and hypothalamus in the regions we profiled so far. And here just shows the correspondence of the 2 different computational methods, shows the robustness of the changes that we observed, number of DE genes versus the Auger score, a very good correspondence. And here shows the major groups of changes in non-neuronal and immature neuronal group. And with the highlights, and as you can see here, all the major non-neuronal types have large number of HDE genes detected. And also further, we use the red color to highlight those non-neuronal types and immature neuronal types that are associated with regions of adult neurogenesis. Okay. And then here is a graph showing all the changes observed in neuronal subclasses. First of all, you see a cloud, a gray cloud of changes in many of the neuronal subclasses. But they all have like kind of subtle, small changes associated with small number of HDE genes. At the same time, we highlight a small group of neuronal subclasses that exhibit really large changes with large number of DE genes associated. And with the red highlighted here, as you can see that many of those special neuronal subclasses are associated with hypothalamus. So here is just a summary of all the changes that we observed in this 18-month-old. It's not really, really old. We now have data on the really old geriatric mice as well, but this is even at this mid-age old mice, those are the kind of many of the changes we observe. So 3 major groups of changes. One is at the neuronal level. We observe decreased adult neurogenesis, decreased neuronal signaling, and decreased neuronal structural integrity. The adult neurogenesis is illustrated by the depletion of immature neuronal types, as well as the change in astrocyte part. And adult neuronal signaling, of course, is associated with changes of many genes important for neuron function, whereas the neuronal structural change is mainly reflected in the deterioration of the long-range axon projections on the neuronal side, and also in oligodendrocytes. We see dramatic changes in oligodendrocyte populations, especially in the hindbrain, brainstem areas, that strongly indicate the deterioration of long-range axon projections. And then the second major change is the increased immune response in the brain, in this mid-old age mouse, healthy mouse brain. We see large numbers of genes associated with inflammation and increased immune responses in many different cell types, in particular microglia, border-associated macrophages, and also even in vasculature, cell types involved in vasculature. And finally, we see a hotspot of aging. And that is the hotspot is the combination of deterioration of neuronal function and increased immune and inflammatory responses. And this hotspot is located within the hypothalamus, especially around the third ventricle, involving the tanycytes, ependymal cells lining the third ventricle, as well as a group of neuronal cells that are just right close to the third ventricle that are important for functions, metabolic and homeostatic functions. So here shows that result. This shows the third ventricle, ventral third ventricle within hypothalamus with ependymal cells lining the top part of the ventricle and different types of tanycytes lining the bottom part of ventricles and also the median eminence. And we see large number of gene expression changes more than astrocytes in the tanycyte and ependymal cell populations. And on the right side, showing the set of neuronal types. So here, not only at the subclass level now, we are able to identify a high degree of gene expression changes in specific clusters. As highlighted is showing this, each column is a particular cluster. The blue ones showing the number of HDE genes. And all these different neuronal clusters are concentrated in those areas that are close to third ventricle, including arcuate nucleus, dorsal medial hypothalamic nucleus, and a paraventricular nucleus. And all those neuronal clusters express important neuropeptides that are known to be involved in feeding behavior, energy homeostasis, and metabolism. And those include MPY, AGRP, POMC, as well as stress hormones such as CRH and other important neuropeptides such as vasopressin, oxytocin, and also cell types that express leptin receptor, dorsal DMH cell types expressing leptin receptor and the GLP-1 receptor. So I think very strong evidence showing those neuronal and glial populations within the third ventricle hypothalamic areas that are undergoing the greatest age-associated changes. Okay, So with that, I want to also just quickly, one slide, showing the work that we have also begun to do in creating cell atlases in the developing brain. And in this particular example, we again profiled a large number of cells, focusing on one cortical area. This is the mouse visual cortex. We profiled the entire prenatal and postnatal developmental stages in this one region with dense temporal sampling. The reason we wanted to do dense temporal sampling is we want to use just the transcriptomic data to obtain trajectories of each cell type that's detected in adult, without having to do lineage tracing studies and things like that. So we used the just transcriptomic similarity across time. We were able to derive trajectories, which are not shown here, but as you can already see the hints from this UMAP, the age progression. And I just wanted to, and in the future, I don't have time today, but in the future I will talk in more detail about our developmental studies. Here, I just wanted to highlight one very interesting point, and this is the time points, perinatal time points, E18, E18.5 to P0. It's just a transition, and the time point in the UMAP is shown right here. As you can see, from E18 to P0. So before that is prenatal time points, is when the neurogenesis and even some of the gliogenesis happens, where all the major subclasses of cell types form in the cortex. But after the pup is born, after P0, it's all neuronal differentiation because neurogenesis has pretty much completed. And what you can see amazingly here is the tremendous diversification during the postnatal differentiation process that can be now precisely timed with the neuronal maturation, synapse formation, circuit interaction, incoming axon projections, incoming sensory signals, neuronal activities, all these things that happens that allows the formation of the adult mature visual circuits is associated with the dramatically expanded diversity of the single cell transcriptomic profiles. So, so it's amazing to see that many of the mature neuronal types are actually formed, emerged during the postnatal differentiation process, probably because of this interaction within the circuit, beyond the initial neurogenesis. Okay, and then finally, a couple of slides showing our work in trying to understand, really begin to understand how transcriptomic profiles are related to connectivity and things like that. And this is one example showing one study to perform this kind of a whole brain full neuronal morphology reconstruction to obtain the long-range projections of individual neurons. And using genetic tools to define the specific types. So in this particular study, we reconstructed 1,700 neurons morphologies, including cortical cells, thalamic cells, and striatal cells. And from this, we find highly distinct projection patterns among the major molecularly defined cell types, which is great. That is confirmatory, not surprising, but it also shows that our molecularly defined cell types are on the right track because the projection patterns are very distinct. But then on top of that, within each of the molecularly defined cell types, we observe further heterogeneity and diversity at the single cell level. And that further heterogeneity is reflected in three aspects. There's additional regional specificity. Even if a molecular type is cross regions, the projection patterns would be regional specific. And on top of region specific, there's topographic specificity. Even within the same brain regions, there are topographic gradients, and that's reflected in the long-range projection patterns, even though the molecular identities of cells may be similar, or maybe they are encoded by subtler molecular gradients within a particular transcriptomically defined type, right? And finally, individual cell variability. Every cell has a unique subset of projection targets, compared to the population. So there's even more, just wanted to illustrate here that there's even more heterogeneity among the individual cells within a particular molecularly defined type. And we use the patch-seq. This shows the patch-seq study, which we want to perform this kind of multi-model classification of cell types, and we focus on the GABAergic interneuron types within the mouse visual cortex. So in this study, we profiled about 1,500 cells getting electrical and transcriptomic signatures, and 500 cells with morphology. And we generated this MET types, the triple modality integrated types. And those MET, because of the heterogeneity of the properties among all these different modalities, we grouped them into 28 MET types, which have lower level of granularity compared to 61 T-types or transcriptomic types that we obtained. So, but those MET types, even though they have intermediate granularity, they have higher cross-modality correspondence and mutual predictability. So we think by grouping the cells into a smaller number of types, it gives us more consistency across the multiple data modalities. And those MET types recapitulate traditionally defined GABAergic interneuron types, as shown by the numbers, by the names available here. Those are all the traditional names, such as basket cells, chandelier cells, Martinelli cells, and VIP positive bipolar cells and things like that. So they are very consistent with traditionally defined, and with further subdivisions driven by cortical layer specificity. A beautiful example shown here, the somatostatin positive interneuron types that are distributed, the MET types that are really distributed from layer 2, 3 all the way down to layer 6 in a layer specific manner. And then ultimately we believe that the MET types, MET types, can serve as a Rosetta Stone to bridge molecular and morphoelectrical properties. And this is shown in my last data slide here, in which that we're now trying to interpret the results that we generated from the large-scale electron microscopy data set that we obtained. We reconstructed the individual neurons and their local synaptic connection properties, but we want to know how those individual neurons reconstructed from the EM data set correlate with the transcriptomic cell types that we defined. So then we use the MET type as a bridge to link the two together. And here is a slide, an example showing that we defined the layer 5 somatostatin interneuron types using the patch-seq data. And through the common morphology matching and the machine learning approach based on morphology matching, we're able to link the transcriptomic types with neurons reconstructed from the EM data, and then associated transcriptomic types with very, very specific, actually interestingly specific synaptic connections between those somatostatin cells and its neighboring glutamate excitatory neurons. Okay, so with that, I just want to summarize with all this huge body of work that's done. Our ultimate goal is to bridge cell types and brain function. We want to do that through the creation of a brain-wide cell type map across 4 modalities, across species. Pretty much all the work I told you about is from mouse, but now we are also doing similar work in human and non-human primate brains. And across modalities, again, the bulk of my talk is really focusing on transcriptomics, epigenomics, and spatial transcriptomics. But there's so much more that need to be done and now can be done into different modalities, different cellular properties, and relate them, integrate them together. And across space, we look at the brain. We don't look at one single region at a time. We look at the whole brain because the circuits are spanning the different brain regions, and different brain regions really work together to generate behavior. And there are all these similarities and differences across different regions. And finally, across time. From development to aging, we want to see how cell types change, and also how cell types change under functional and diseased conditions. Okay, so with that, I just wanted to say in my talk today, I didn't talk much about the spatial work. But going forward, spatial biology is going to be tremendously important in all these different aspects, as you know. We are also now using not only the Murphy platform, but also 10x Xenium platform, the StereoSeq-based sequencing platform, as well as getting into spatial proteomics as well, deepening our understanding of cell type diversity in the brain. Okay, so with that, I think I'm running out of time, so I want to just, I don't have to reiterate the summary conclusions from what you told about. They are shown here. And I want to thank a large team of scientists and investigators at the Allen Institute and all the project leads of the different projects that I told you about. It's really, really this kind of a collective team effort that allows this kind of a comprehensive work to happen and allow the insights to be generated from the work. The combination of experimentalists, computational scientists together is really amazing to see how much we can achieve. Okay, thank you very much for your attention. If I have time, maybe a few questions. Yeah, thank you very much. Sorry about the long talk.
Yeah. I mean, what a course work.
Thank you.
It's always fascinating to see how complicated and how much information is still learning from the mouse brain.
Yeah, it's complicated, but now we have the means to dissect that complexity.
Oh, sorry. Yeah. Well, thank you. I mean, this has been really, I was really looking so much forward to this talk. So I train in Spain, and I used to visit Instituto Cajal very often and, look at the actual original printings. And I can't keep thinking how amazed Cajal himself would be if he would be sitting today here. So this is really how far we have come from what he was, kind of trying to see. And what we can see now is just absolutely amazing.
Thank you.
I had a particular question. So your data on the aging of hypothalamus, I think, has potential very important implications in how we interpret human aging. So, hypothalamus is circadian rhythm, sleep, hormone regulation. We have all of that cohort of homeostasis that is running pretty much from that nucleus that derives pretty much our daily functions. And so I wonder if you think that human aging and disturbances of sleep and many other, normal, essentially comorbidities that happen throughout our normal aging could be actually driven primarily from the aging of the hypothalamic regions that you are seeing in your models, and whether therefore targeting that region could be a potential new way to prevent or modify some of these comorbidities that then impact the majority of the diseases we study, such as neurodegenerative and so on.
Yeah, that's a great question. I think you're specifically asking about aging, right? Yeah, I think so. I think a lot of the aging effect, they could have a common source, and that could be really is the brain-body interactions and the metabolic changes that then would affect so many other aspects of that. So I think this kind of comprehensive profiling is really important, and maybe time trajectory as well. It's harder to do it in human because there's all this heterogeneity, variability, and all things like that. But animal models, I think, yeah, it's a very good starting point to think about that in this kind of a global view about how aging is happening in a coordinating manner. Yeah.
Hi. I think you were talking a little bit about the midbrain, and you're talking about some populations of neurons that were spatially mingled and co-expressing multiple types of signaling machinery.
Yeah.
So I was wondering, can you talk a little bit more about what these cells are doing and what's governing their function?
Yeah, so the midbrain part, you're probably talking about the dopamine neurons. Right? I specifically use the midbrain dopamine neuron as an example. Dopamine neurons are very important, for reward and motivation. In the brain. So it's a very important neuromodulatory transmitters that drives the motivational behavior and all the kind of reward aspects. The heterogeneity was suspected before, but this kind of heterogeneity is really kind of unexpected, and the intermingle of the different kinds of dopamine neurons together. There was one kind of a uniform theory about how dopamine neurons mediate this kind of reward. It's called a prediction error signal. But we now know that hypothesis, the prediction error model for the dopamine neurons is probably going to be outdated because of the heterogeneity of the dopamine neurons that we observe. And now there's also increasing physiological evidence suggesting that the responses of the dopamine neurons under a particular behavior condition is actually quite heterogeneous. So, yeah. And that's just one example. There are many other brain regions have multiple cell types intermingled together as well. Very much like you study cancer, immunity with the tissue, it's very heterogeneous cell types that are interacting very closely together within a particular region. Yeah.
Hey, I have a question here in the back. Really fantastic work. Thank you very much. I'm wondering if on the aging aspect of your analysis, how well do you think the observations from the mice translate to humans, given the huge difference in lifespan?
Yeah, of course, it's hard to translate. This is inbred mice under a kind of a restricted housing condition. It needs to be validated, but I think the general biological process, aging process, is pretty similar, right? Even the time course is different, but yeah. So we think it's generally translatable, but of course we'll need more data to be able to translate. And I hope that more species can be profiled, and also maybe mice in the wild conditions, wild mice may be better in that aspect as well. But importantly, is cross-species comparative study would be essential.